paper we published in 2019, and it was entitled Spironolactone depletes the XPB protein and inhibits DNA damage responses in UVB irradiated human skin. And so spironolactone is an FDA-approved drug. It's used for a couple of purposes. It's a mineralocorticoid receptor antagonist, so it can be used to regulate things like blood pressure, and it's used for example, in heart failure patients. It's also an androgen receptor antagonist, and so it can be used to, most commonly used for in adult women with acne to control that skin condition. Again, typically taken as an oral medication, although there have been some studies developing as a topical compound. However, in 2014, a group in France was screening FDA-approved drugs to look at things that inhibit nucleotide excision repair. Nucleotide excision repair is the main system, or the only system in human cells that can remove UV photoproducts from DNA. It also removes other things like cisplatin, so it's relevant for cancer therapy, in addition to, for example, skin cancer risk. However, this group in 2014 showed that actually spironolactone inhibited nucleotide excision repair, so photoproducts from DNA. And they show subsequently that it leads to a rapid degradation through the proteasome of a protein known as XPB. So XPB is essential for nucleotide excision repair. It's mutated in a subset of patients with this disease, xeroderma pigmentosum. And so we were interested in, okay, it does something like this happen in the context of human skin. So we initially started with simply keratinocytes in vitro. So we took telomerase immortalized neonatal foreskin keratinocytes, exposed different doses of spironolactone for, I believe this is 2 hours, and then harvested the cells and made protein lysates. And so, band indicated by this asterisk, which is not XPB. However, if we look at the XPB protein levels, we see a nice dose-dependent decrease in XPB protein levels. In Figure 1b, it's simply a time course, and so this is consistent with other publications that have used other cell lines, but in keratinocytes as well, hours or so, you can almost completely remove XPB from these cells. Now, consistent with the idea that XPB is important for nucleotide excision repair, if you treat cells with spironolactone and do immunodot blotting, so this is where we take genomic DNA from these cells at different time points after UV radiation under these different conditions, vehicle or spironolactone, and then immobilize that DNA on a membrane and then probe with antibodies against either the CPD, a major UV photoproduct, or total DNA. We can see the spironolactone essentially completely inhibits, or nearly completely inhibits, the removal of CPDs from DNA. We've also looked at sort of additional control. So we know that during nucleotide excision repair that generates gaps in DNA once the damage is removed. And one of the ways we can quantify or measure the gap presence is by an enrichment of RPA on total chromatin of cells. So after DNA damage, you get gaps. It becomes bound by a protein known as RPA, which is a single-stranded DNA binding protein. So in the vehicle-treated cells, we can see that total levels of RPA on the chromatin fraction of the cells, so what's bound to DNA and histones, increases after UV exposure. But cells treated with spironolactone, that is completely abrogated. So there's essentially no repair, or very little repair taking place, or little gaps being generated. Now, we also know that these gaps are signals for activating DNA response kinases, including ATR and ATM. And so two of the substrates of these proteins include CAP1, a heterochromatin protein, P53, a tumor suppressor protein. And we can see that in these cells, these are asynchronously growing, treatment with spironolactone does reduce partially the level of phosphorylation of these kinases in these cells. From other data, if we use cells in quiescent cells, in a quiescent state, non-growing replicating state, we can actually very more significantly reduce this level of phosphorylation. However, when you have S-phase cells, the signaling pathways that activate these kinases are a little bit different. Nonetheless, we can partially inhibit some of this DNA damage kinase signaling by treatment of keratinocytes in vitro with spironolactone. Now, consistent with the idea that you need repair for cell survival, spironolactone using an MTT assay, which measures acute cell survival. This is maybe a different repair assay or a survival assay. Nonetheless, spironolactone significantly reduces the level of the survival of these cells. So again, this was with telomerase immortalized keratinocytes. Is this relevant for normal keratinocytes, particularly adult keratinocytes? So what we did was to grow primary cultures of adult keratinocytes that were obtained from abdominoplasty skin samples. So this is normal human keratinocyte, and it's just from three different individuals. So in all three cases, spironolactone reduces the level of XPB expression, and it also reduces the level of phosphorylation of these DNA damage response kinase substrates. And then if we look at repair, again, spironolactone inhibits CPD removal and leads to reduced cell survival. So very consistent with what we observed in the telomerase immortalized keratinocytes. So in figure 3, what we want to do is actually look at this in the context of human skin ex vivo. So what we did here is to take, we get abdominoplasty skin samples that are discarded during normal surgeries, and then we can maintain the small punch biopsies of that skin inside a little hanging well insert in a cell culture dish where we have some medium below the biopsy to essentially feed the skin and sort of mimic a blood supply to some extent. And then what we could do is then add spironolactone to that cell culture medium and see if it similarly affects XPB expression. So in these experiments, we added 20 micromolar spironolactone on 3 subsequent days, and then harvested the skin and specifically the epidermis to look at XPB protein levels. And in this particular case, we almost, in this particular sample, almost completely remove spironolactone from spironolactone can almost completely lead to a loss of XPB expression in the epidermis. There is some variability. So in the graph over here, this is from several different skin samples. And so there is some variability from person to person, probably not too surprising. We used another compound that also inhibits the mineralocorticoid receptor. This is eplerenone. This is in the same family as spironolactone, but it does not affect XPB expression. That was previously shown by the French group in 2014. We also did time courses, or dose responses here, looking at increasing doses of spironolactone. Again, in figure 3A there, we used 20 micromolar at this range. So but if we use lower doses, we do see a partial reduction. Again, it is dose dependent. One of the things to point out is this is a very high level of the drug. The peak level present, the peak serum levels found in patients taking a normal dose of spironolactone has been reported to be under 200 nanomolar, and so it'd be down in this range. So again, it's a very little change and not statistically significant from different than the vehicle control. So the relevance of this to actually people taking the drug might be somewhat questioned and minimal. But one of the problems with issues potentially with taking any drug is, what level of repair would be potentially enough to raise skin cancer risk. So those are questions that still need to be addressed. Nonetheless, again, it does take a significant amount of spironolactone to completely remove XPB. Nonetheless, this is potentially a pharmacological way of making knockout skin in human skin. So now, consistent with the idea that we need XPB for DNA repair in the context of the skin epidermis, if we do CPD immunodot blotting to look at repair in a vehicle control skin, we can almost, in many cases, completely remove all the CPDs by 2 hours after UV exposure, by 48 hours or 2 days after UV exposure. But then the skin treated with spironolactone for 3 days, that is completely blocked. Now, consistent with the DNA damage kinase signal we looked at previously in the cells in vitro, treatment with spironolactone can inhibit the DNA damage kinase signaling. And it looks more a bigger block than we see in the proliferating cells in vitro. And that's probably because if you think about total epidermis, very few of those cells are actually replicating. And so we actually believe that the DNA damage kinase signaling that takes place in differentiated cells or quiescent cells in the skin are probably more dependent upon nucleotide excision repair. Now, consistent with the reduced repair and the reduced DNA damage response, we can also look at apoptosis in the skin, and this is by using H&E staining. And this a classical way we can look at sunburn cells. So if you look very closely here, it may be hard to see on the screen. The staining of epidermal keratinocytes looks different in cells that are undergoing cell death, and so you see a little more pink cytosolic staining, and then the nuclei become a little bit more compact. So these are classically called sunburn cells, and so one of the things we could do by looking at a microscope of H&E stained section is to quantify the number of sunburn cells. So we see in a vehicle-treated cell or vehicle-treated skin, we do see some level of sunburn after UV exposure, but it's elevated in the skin treated with spironolactone. And so that's consistent with what we see in vitro. Now, spironolactone is actually metabolized to a number of different metabolites, including canrenone and thiomethylspironolactone. And so these have been measured previously in the skin, and they're actually much more abundant than spironolactone within a few hours after taking the drug. These compounds do have activity towards the mineralocorticoid receptor, for example. And however, we looked at in the polymerase hemolyzed keratinocytes, these compounds essentially have no effect on XPB protein levels, so they seem to be inactive, and that maybe even adds more potentially in terms of the physiological relevance of this drug towards XPB levels in the body. Because it gets metabolized to compounds that don't have this effect, the drug the parent compound, although it can't have this effect because of the metabolism, it's less likely to impact XPB expression potentially. So again, these compounds, the metabolites, do not affect XPB expression. We do look at repair by CPD immunoblotting, and only spironolactone affects repair. Eplerenone, again, is another family member in this compound. It's not a metabolite, but another similarly structured compound. So it seems specific to spironolactone. Again, only spironolactone seems to affect repair or relative survival. And then if we look at mutagenesis, so this is looking at what we call an HPRT assay, looking for cells that become resistant to a drug called 6-thioguanine. So normally cells are sensitive to this drug because they have a wild-type HPRT gene and locus. However, if there's mutations in that gene, the cells become resistant to 6-thioguanine, and they'll grow in the presence of the drug. So we know that UV can induce some of this, and so in a vehicle-treated cells, DMSO, we see about 10 mutants cell become colonies after a few weeks in the presence of 6-thioguanine in per million cells. Spironolactone increases the level of mutagenesis in the cells, increases the number of mutant cells that emerge. However, neither canrenone or eplerenone have that effect. So it seems to be specific to, again, the parent compound. So again, this is again just a brief summary of what we did in this paper. I think the big take-home message is that this drug can affect XPB levels in human skin. The relevance to people taking the drug is reasonably questionable. I don't think most people taking this drug would have much to worry about. There may be people, if they take higher doses of the drug, potentially it may more significantly affect XPB levels. However, if your XPB was completely gone, you'd be very sensitive to the sun. People with mutations in XPB have severe photosensitivity in addition to skin cancer, elevated skin cancer risk. And so most people taking this drug will not likely have any significant effect, noticeable effect in the skin. And indeed, there's no evidence from epidemiology or any other previous reports that people taking this drug have any higher rates of skin cancer. And so, again, there may be very little risk to worry about. However, there have been studies to try to develop topical application of this drug, and this is relevant for things like acne and potentially hair loss, where maybe modulating androgen signaling can be beneficial. But oftentimes when you have a drug that used topically, it's used at much higher concentration, and that might raise some concerns. If you're using higher concentrations of this drug, specifically targeted towards the epidermis, that might become a problem. And that's one of the things we're trying to follow up on to see if that is indeed the case, using higher concentrations that are being tested in patients to see if it does affect XPB levels. Nonetheless, another take-home message from this paper is that this drug can be used actually to generate essentially an XP-like phenotype in a human skin sample. So that's it's not, for most of us, not readily available to get skin from XP patients. This is obviously a very severe disease, and so potentially using this drug could be a way to model that disease state in an ex vivo human system. So that's one of the other, I think, advantages or take-home messages from this paper, is that we could potentially use this drug for that purpose.